Good day, everyone. This is Dr. Faundo from the Stem Cell Center of St. Luke's Medical Center in Global City. I'll be talking about the current applications of stem cell testing. Our objectives for this lecture would be first to review the different types of stem cells and their unique properties. Next, to present current tests that are performed on this different type of stem cells. And third, to discuss future developments in the field. Just a short review of the hierarchy of stem cells. We know that they start as totipotent stem cells, like in the embryo, which are able to give rise to all the different types of organs. Down the line would be pluripotent stem cells, which gives rise to many types of cells and organs, foremost among which are hematopoietic stem cells that give rise to our different blood cells, like red cells and white cells. And on the other hand, would be the mesenchymal stem cells, which form the different supporting structures like muscle, nerve, bone, and other tissues. So for this lecture, I will be focusing the talk on hematopoietic stem cells, or HSC, and mesenchymal stem cells, or MSC, since these are the two most common types that we are working on in the country. So where can stem cells be obtained? First would be the embryo. As mentioned, it contains totipotent stem cells. So these are actually the most ideal stem cells to work on. However, these are prohibited in the Philippines. Next type would be the perinatal tissues, which are obtained at birth. And this comprises the placenta, the umbilical cord, and the amniotic fluid. So obviously, these perinatal tissues are discarded as waste, but they have been found to contain stem cells which may be useful for future applications. However, most of us would not have our perinatal tissues stored when we were born. So for practical purposes, our patients nowadays would usually resort to adult sources of stem cells, and there are many of them. So the different adult sources of stem cells would first be the bone marrow. So obviously this contains a lot of stem cells since they are required to produce our blood cells throughout life. Most of what we know about stem cells are based on studies from the bone marrow, studies way back in the 50s and 60s. So this is not something new. However, in the 90s, we were able to obtain stem cells from the circulating blood and collected through apheresis. And this is probably the most common way of collecting stem cells nowadays in the country. On the other hand, many other workers are using stem cells coming from other sources like adipose tissue as a product of liposuction procedures. The ophthalmologist can get stem cells from the limbic area, or the orthopedic surgeons can get stem cells from the joint. Cardiologists are working on stem cells coming from the heart itself, and may even obtain stem cells from deciduous teeth. Okay, so there are many sources of adult stem cells. So what is the status in our country? Back in 2013, the Department of Health issued administrative order in order to regulate and to accredit health facilities that are engaged in stem cell and cell-based or cellular therapies. And take note that in this order, it is stated that the laboratory shall be headed by a board-certified pathologist. And that is the reason we are discussing this, since we would like to keep that way, wherein the laboratories are being headed and managed by pathologists rather than someone outside of our specialty. I would just like to point out in a particular portion of the administrative order, the list of the stem cell preparations that are registered and permitted. I have mentioned that use of embryonic stem cells is prohibited in our country for ethical reasons. So what is permitted would be autologous stem cells coming from the adult. And the word autologous means that this comes from the same patient and may undergo basic processing like separation, fractionation, primary culture, and expansion, but without modification genetically. And this same stem cells would be given back to the patient. The other general type would be allogeneic, meaning it comes from a donor. Again, it may be followed by basic processing, as mentioned earlier, and without genetic modification. For allogeneic sources, HLA typing would be required, obviously for hematopoietic purposes, as well as infectious disease screening, like that, what we use for blood transfusions. So this includes hepatitis B, hepatitis C, HIV, etc. So what are the tests that are performed on stem cells? So I'll be showing you here the most common ones that we are performing, and this includes cell count, viability, sterility, and potency. So for stem cell count, we now rely on a flow cytometer to do the job. Back in the days, a total nucleated count, which includes all the nucleated population, would suffice for bone marrow harvests. But nowadays, we are using CD34 counting for hematopoietic stem cells, especially for those obtained by apheresis or from cord blood. So for hematopoietic transplants, we already know the usual dosage, which is about 3 to 5 million per kilogram of body weight. So it is dependent on the weight of the recipient. Meaning, if you have a small donor that's going to give to a bigger recipient, then you will have to collect more, even on succeeding or multiple days. On the other hand, if you are working with mesenchymal stem cells, the markers that are used are CD73, CD90, and CD105. These are the markers accepted by the International Society for Cell and Gene Therapy. For mesenchymal stem cell applications, these are mostly for experimental or clinical trial use only. So we do not know yet what is the ideal dosage and even the ideal route of administration. So current studies would administer mesenchymal stem cells intravenously. It can be injected directly to the affected organ. So some are doing it intrathecally or direct into the joint space, or by any other means. So again, For hematopoietic stem cells, we already know the required dosage, but for mesenchymal stem cells, these are still being studied. Now, after knowing the stem cell count, we would like to know if the cells are viable because we would like them to be alive when we give it back to the patient. And there are two general methods of performing viability. One is manual and the other is automated. For manual testing, we generally use a reagent called trypan blue. And this reagent will enter the cell if the cell membrane is damaged, such that the cells that become blue are dead because the membrane has allowed it to get inside the cell. And by counting the number of live cells, meaning those did not take up the dye, over the total number of cells, then we are able to compute for the viability. So again, this is not a direct measure of the cells being alive, but it is a measure of the integrity of the cell membrane that is used to assess the viability. The other method is the use of the reagent 7-AAD, and this is performed in a flow cytometer. So usually done at the same time when the cell count is performed. So this reagent will attach to the live cells. So the live cells over the total number of cells will give you the percent viability. So those are the two common methods. Next, we would like to check if the stem cells do not have any contamination. So for hematopoietic stem cell products, we would perform aerobic and anaerobic culture. It can be done at collection or after the infusion of the stem cells, depending on the protocol. Okay. So for example, in apheresis collection, it may be necessary to do a culture just to ensure that there are no organisms that are obtained during the procedure, since they usually make use of large bore catheters that are inserted through the femoral vessels. So you can imagine the possibility of contamination coming from the normal flora in that region. However, if the techniques have been optimized, then it may not be done regularly, but only as needed or at regular intervals. At the other end, we can also perform a sterility testing after we have infused the stem cells in order to check if there are any organisms that may have contaminated the product from the time it was collected, processed, stored, and infused to the recipient. At that point, if there is anything that would show up, then the patient is treated based on the sensitivity studies. However, please note that most of recipients of hematopoietic stem cells will already receive prophylactic antibiotic coverage. Next is fungal culture. Again, it is very possible that the stem cell products would have fungal contamination, and you can imagine this happening, for example, in a bone marrow harvest, wherein multiple punctures are done on the iliac crest to obtain a bone marrow sample that can amount to up to a liter in large donors. So there is that possibility of some type of contamination, whether bacterial or fungal. So again, fungal culture may be obtained, and if there is anything that grows, then the recipient will be treated based on the results. Next, for those stem cells that will have to undergo culture, then mycoplasma testing will have to be performed, since you know that this is a very common contaminant in cell cultures. And I will not go into the different methodologies here, but suffice to say that it is required for stem cells that have undergone culture. And lastly, endotoxin testing would be required again for those stem cells that have undergone culture, since you know that the endotoxins may be present even if the organism is not present, and they are stable even with the application of heat. So testing will have to be performed to ensure that no endotoxins have been introduced during the multiple passages. So before we proceed, I would just like to bring up the principle of cryopreservation. So in cases wherein stem cells will have to be collected, processed, and then stored for use sometime in the near future, or even many years later, then the stem cells will need to be cryopreserved. Otherwise, if you put them at a temperature of minus 150 Celsius, then you expect the cells to shrink and die. So what we do is add a cryoprotective agent, most commonly dimethyl sulfoxide or DMSO. And this reagent provides a hyperosmotic environment around the cells, preventing them from shrinking as they are placed in a cold environment. So this is the general principle of cryopreservation. So we do this usually for those patients who have autologous transplants, and even for those who will undergo allogeneic transplants, but the donor cannot be around at a specific time. So we can harvest the donor stem cells and cryopreserve them. Or, for example, if the donor is located remotely, so the stem cells can be collected from the donor and then sent to another facility for processing and storage so that it can be used on the recipient at some other time. So just to show here the schema of what we call an autologous transplant, and transplant in quotation marks, since this actually comes from the same patient and given back to that same patient. So a patient who is sick, for example, with lymphoma and is put into remission by chemotherapy will have his stem cells harvested, and the stem cells will now be set aside, as shown in the gray box here, and cryopreserved, so that when this same patient will undergo chemotherapy or irradiation, then the stem cells are preserved or protected from the effects of chemotherapy and radiation. And at some point, when the patient is ready for a transplant, then the stem cells are thawed from a temperature of minus 150 Celsius to a body temperature of 37 degrees and returned to the patient. So this is also called as a stem cell rescue, because the high dose chemotherapy and the radiation that may be given will destroy almost the entire bone marrow of the patient. So giving back the patient's own bone marrow will give the patient a chance to regenerate his bone marrow. Okay, so In here, the stem cells are not the ones that are used for the cure, but instead it is still the chemotherapy and radiation that will kill the main bulk of the tumor, but at the same time destroy the patient's bone marrow. So the stem cells here are just done to rescue the bone marrow. So having said that, the other important test that will have to be done would be to test the potency and characterization of the stem cells. So the most common way would be to culture the cells, just like the way we culture organisms. But here, what we see are colony forming units. So each type of hematopoietic lineage will exhibit particular appearances on a plate. So whether you are looking for erythroid precursors or granulocytic precursors or megakaryocytic precursors, then these are counted, and these are then assessed to check if the harvested stem cells would be able to grow in vitro. And this is used as a measure to check for their potency, meaning that the stem cells that have been given would be expected to grow. And that gives us a measure that the processing and the storage did not harm the stem cells. Take note that this takes weeks in order to get results. So the stem cells might have been transplanted already, and the cell culture results will come in many weeks after the transplant. So this is more of a quality measure rather than to give a decision whether to give the stem cells or not. So right now, there are still many other researchers looking on other ways to have these results in a much shorter period of time, such that appropriate action may still be performed in case you get a negative finding or cells which are not proliferating properly. Next, if you're working with mesenchymal stem cells, for example, then a proliferation activity will have to be done in order to ensure that the stem cells you're working with will actually grow into the type that you want them to become. So for example, with mesenchymal stem cells, you would like them to have the morphology of spindle cells like what we see here, and that shows you that they are actually growing along that lineage. Or if you are working with epithelial stem cells, for example, like regeneration of the pancreas or the liver, then you would want to ensure that they are actually becoming epithelial cells and not fibroblasts. I have not shown here, but you may also perform after this markers to ensure that they are going down the line that you want them to become. So if you are trying to grow liver cells, then you can mark them with markers, for example, for albumin, okay, and for other specific type of cells. If you're working with neural cells, then you attach neural markers in order to characterize the type of cells you are working with. So the cell therapy field is really expanding, and we just mentioned about stem cells. So it is actually a conglomeration of different fields. So stem cell therapy, as I mentioned, started with bone marrow harvests back in the 60s, going all the way to the 70s and 80s. Okay, so most textbooks would still call it a bone marrow transplant, since that forms the basis of what we know about stem cell transplant right now. And nowadays, there are also what we call gene therapies to treat specifically monogenic diseases. And the examples would be sickle cell, anemia, and beta thalassemia. So what is done here would be stem cells from the patient would have the correct, the defective genes corrected and given back to them, even in childhood or even in utero. So these are all the gene therapy studies that are now ongoing. And more recently, also hemophilia A and B. Aside from that, the stem cells are also used now for cell-based drug delivery. You know that sometimes if you give systemic drug treatment, then you would expect side effects to occur aside from your intended effects. But if you are able to attach the drug to the cell and deliver it to the exact site where you want it to perform its job, then a cell-based drug delivery would be ideal. I'm sure you have heard about cancer vaccine. So in this case, the cells are reacted against tumor cells so that this becomes sensitized, and your own immune cells, for example, will now act versus your own cancer cells. And you've probably heard about regenerative medicine. So this is a field that covers many areas, and basically a type of therapy that addresses those conditions that are caused by degenerative changes. And these include osteoarthritis, Alzheimer, Parkinson's, and many other types of conditions that affect the heart, the brain, spinal cord, etc. And lastly, stem cells are also being used in tissue engineering. And basically, this is done by growing cells into the type that you want them to be. For example, hepatocytes, okay, and making them grow on a scaffold so that these are easily transplantable. You know that it's not easy to get transplants from donors. So what is being studied here is the possibility of growing your own cells and transplanting it back to the person who has a diseased organ that is damaged by cancer or trauma or other conditions. And even in the pharmaceutical industry, stem cells are also used for toxicity testing. So instead of using animal models for toxicity testing, it is believed that stem cells can be used in the future to test for the drug effects, as well as to test for any toxicity that may be associated with that particular drug. Or you can start with stem cells and make them differentiate into different types to generate neurons, blood cells, or liver cells, for example. And this can be used to study the effects of drugs and even to understand how to prevent and treat birth defects that may be associated with drugs. So this really will push the role of stem cells because you will do away with using animal models and even doing many of the clinical trials that involves human subjects. So what does the future hold? There is a continuous discovery of new sources of stem cells, aside from that I have mentioned earlier. And of course, with the discovery of these new sources would be the need to check if they are indeed stem cells and they are able to perform their functions. Aside from that, once the new sources are discovered, then new methods will have to come in order to acquire them and to process them, to harvest. And of course, testing is an integral part of all these endeavors, because you would like to ensure that you're actually getting stem cells and not mature or fully differentiated cells that may not be useful for your purpose. And with the increasing incidence of chronic diseases in an elderly population, there is a rising demand for regenerative medicine. So the idea here is that stem cells could be collected from a person, and they may be processed and stored such that they can be given back to that same person in the future if there is a need to uh, to correct a particular condition that affects the nervous system, the heart, the joints, etc. So in all of this, let us remember that the laboratory plays a role in the testing for all these different types of stem cells in order to ensure that you have enough of these cells, that they are going to perform as expected, and as well as testing after they have been administered in order to check their potency as well as the general effectiveness. So, what did we learn today? So, as a take-home message, I hope that I have been able to convey to you that the stem cells can be obtained from multiple sources, and they have their own unique properties that may affect how they are going to be used in the future. And being laboratory people, we are concerned with the tests that are performed to ensure that there is adequate stem cells that are collected, that they are viable for use, that they are free from any types of contamination, and that they are potent and are able to perform or function as expected of them. And lastly, I hope that I was able to make you see that the stem cell field is rapidly expanding with newer challenges in the future. And I hope that some of you will consider going into this field, since it is still a very young field, and many things are still unknown to many of us, so that a lot of researches would still be needed in this field. So that is my last slide, and thank you for your attention.